Good day, everyone. Mr. Mechnick with a podcast on buffers. So here's the goal. Let's just try to understand chemically what makes a buffer and why they are important for living organisms. So, a buffer is simply a solution that contains a weak acid and a conjugate base. And both of these work together to resist changes in their pH. So they can allow the pH to be stable. So ultimately, different buffers work in different pH ranges, and they're kind of adapted to the host that they're found in or the environment they're created for. So when you have this weak acid and conjugate base working together, the acid can donate protons, and the weak base, can accept them. So as a result, there's a giving and a taking of these hydrogen ions. So when we look at examples of buffers in living systems, the two most common ones are the phosphate buffering system. So right here, H2PO4. This is the weak acid, and this is the conjugate base. So as you can see, H2PO4 has the ability to donate hydrogen ions, and when this happens, it will drop the pH, keeping it at that stable level. HPO4 has the ability to accept hydrogen ions, and as a result, it can create H2PO4. So H2PO4 is the weak acid, HPO4 is the conjugate base. This is the donor, this is the acceptor. Here's another common buffer found in living things. It's called the carbonate buffer system. H2CO3 can dissociate into H plus ions and HCO3. So this is the conjugate base, this is the weak acid. The weak acid can donate hydrogen ions to the solution, and HCO3 can accept hydrogen ions. So what makes a weak acid useful for buffers? Well, first thing we should understand, all of these have to involve water at some way. Why can't we just have water acting as our buffer? Because water is a very, very, very weak acid. So when you have H2O molecules, they can dissociate, but it rarely happens. Only two out of every billion molecules of pure water will ionize or dissociate into hydronium and hydroxide ions. So because of that fact, because water doesn't dissociate that often, it's a very weak acid and would not be good at counteracting the effects of the addition of H's or OH's to a solution. So what you end up needing is something that is a little bit better at dissociating when it's in a solution. So some substances like acetic acid or vinegar dissociates very poorly. So even though acetic acid is an acid, it's a very weak acid because when it's mixed with water, it partially dissociates into CH3COO and some hydronium. So the hydronium is what makes this acidic. And this conjugate base has the ability to accept that to make the acetic acid. And we can't use something like nitric acid, HNO3, because this stuff, when you put it into solution, is very aggressive and will fully dissociate. So the HNO3, when mixed with water, will fully dissociate into hydronium and nitric oxide. So this is not good to act as a buffer. So we need something that's kind of in between, and that's where some of these substances that are found in living systems work well. So we can't use a strong acid. We can't use water because it's way too weak to ionize. So we need the weak acid that's just right. We need something that can partially dissociate when it's put into a solution. So if we take a weak acid, it will partially dissociate, and it should have that conjugate base, which can accept those protons as well. So weak acids only have a modest tendency to shed their protons. And when they do this, the negatively charged anion becomes a willing proton acceptor, and that's the conjugate base. So the conjugate base has the ability to accept those protons. So in reality, what's very important for you to understand is that strong acids don't act as good buffers because they completely dissociate. So these little red dots, those are all the H ions, which makes the solution very acidic. Where you have a weak acid, so some of these dissociate, so weak acids do much better job acting as buffers. When you have a buffer solution, every buffer can be busted, so to speak. What that means is we can go beyond the capacity of that buffer. So the buffer only has a certain limit. So the limit that we kind of often refer to when we talk about buffers is this equilibrium point. If you take an AP chemistry class, you'll learn about this pK equilibrium calculation that you can use to actually determine what the pK value would be for a specific buffer solution. But if you look at this graph, what it's basically saying, if we add a bunch of acid to this initial solution, we can see that the more acid you add, eventually it's going to get to a point and it's going to drop very rapidly. Or on the other hand, you can add a base to this solution and the pH will start to climb and it will get to a point here where the conjugate acid is exhausted and the pH is going to skyrocket. So at these points where the conjugate base or the conjugate acid is exhausted, that means it doesn't have the ability to either accept more hydrogen ions, and the hydrogen ions are going to accumulate and we're going to drop the pH a lot, or we're going to end up having not enough H's to donate anymore, and as a result, the pH is going to skyrocket. So when you see this type of graph, we're looking at this region here where the buffer is actually working. But once it goes beyond its capacity, then the buffer is going to change significantly. So why is this important for living things? Well, as we will learn in the future, proteins play an important role in all living systems. One type of protein that acts as a biological catalyst are called enzymes. These things can be affected significantly by changes in pH. They're not going to function if the pH drops or raises too high. So all living things are made up of cells. Cells are made up of a lot of proteins, and these proteins make your cells work the way they do, and without having the proper pH, your cells will be out of balance. They will not maintain homeostasis. So when you have this buffering capability, it's essential for living things to perform their job. So even though our stomach acid might have a pH of 1, a very, very strong acid to help break down the proteins that we eat, but yet you get into the small intestines and the pH inside those cells is near 7. So the pH in our cells, we need to keep it right around that 7 level so it can function properly. So a lot of these compounds in our body that are essential for life depend upon this bicarbonate buffering system to allow for our cells to function in a very narrow pH range. So our blood, for instance, has a very narrow pH range, right around 7.35, for instance, and we have to keep it right there so our blood contains a protein called hemoglobin can transport oxygen to the cells that require it. Thank you for listening, and please bring with you any questions or uncertainties about buffers that you have. Thank you.